Okay, let's unpack this. Think about it for a second. The food you ate today, whatever it was, is currently undergoing one of the most incredible, intricate, and frankly unseen transformations imaginable inside your body.
It really is.
It's this complex chemical and physical journey that is absolutely fundamental to, well, to you being alive.
It truly is. It's like this continuous process of biological engineering happening around the clock. And mostly, we don't even give it a second thought.
And that's exactly what we're diving deep into today. We're going to explore precisely how your body takes those major food components, the carbohydrates, fats, and proteins, breaks them down piece by piece.
And then masterfully absorbs the crucial nutrients, along with all that essential water and electrolytes. It's quite a feat.
Yeah, we're going to take a really close look at the detailed processes happening right inside your gastrointestinal tract. And we're guided here by some incredibly detailed physiological texts that you provided.
This source material gives us a really solid foundation. It gets right down to the cellular level, even the molecular level, which is exactly where these transformations are orchestrated.
Right. So our mission today is to unpack these concepts so thoroughly that hopefully you gain a level of understanding akin to really mastering the subject.
Getting into the weeds, so to speak.
Excellent. By the end of this deep dive, you'll understand the specific chemical reactions, the key molecular players like the enzymes and transporters, the specialized anatomical structures involved.
And you'll really appreciate the remarkable efficiency that lets your body extract every bit of nourishment.
So let's get started. To really understand digestion, we have to start with the most fundamental chemical process the body uses. It's how it breaks down those large food molecules.
Because the basic challenge is simple, right? The complex carbs, fats, and proteins we eat are just too big.
Way too big. They can't pass directly through the lining of your gut into your bloodstream.
Nope. They need to be chopped up into much smaller pieces first.
Think of it like having to dismantle a large Lego structure.
Or a complex machine, maybe.
Yeah, exactly. Before you can carry the individual bricks or parts through a small doorway.
That's a good analogy.
And the universal tool the body uses for this dismantling is a chemical reaction called hydrolysis.
Hydrolysis, okay.
Now, to really get hydrolysis, it actually helps to first picture the reverse process, which is how these large molecules were built in the first place. That's condensation.
Right. Condensation is how your body, or, plants, build larger molecules from smaller ones. Imagine you have two smaller building blocks.
Okay.
To link them, the body effectively removes a hydrogen atom, an H+, from one molecule, and a hydroxyl group, an OH, from the other.
H+ and OH. Got it.
Those two parts, the H+ and OH, combine perfectly to form a molecule of water, HO. And because those spots were cleared where they were removed, the original two molecules can then link up,
Forming a larger structure.
Exactly, like making a disaccharide from two monosaccharides, or building a triglyceride from glycerol and fatty acids, or linking amino acids to form a protein. Water comes out, things link up.
Right, so hydrolysis is just the complete opposite. Digestion is essentially reversing that condensation by adding water back in.
Okay, putting the water back.
Yeah. Specific enzymes, which are proteins themselves, remember, are secreted into your digestive juices all along the gut.
And these enzymes act as catalysts.
Precisely. They help to return the H+ and OH from a water molecule right back to where they were removed during condensation. When that water molecule splits and adds across the bond,
The bond breaks.
the bond breaks, splitting the larger molecule back into its smaller units. It's like using water as a tiny chemical wedge to pry apart the pieces that were previously linked by removing water.
And this same fundamental chemical trick applies across the board for all the major food types.
Universally. For carbohydrates, large polysaccharides like starch or disaccharides like sucrose and lactose, they get split by hydrolysis into their single unit monosaccharides.
Simple sugars, basically.
Right. For fats, specifically triglycerides, which are formed by condensing 3 fatty acids onto a glycerol molecule, hydrolysis adds those 3 water molecules back in.
Releasing the 3 fatty acids from the glycerol backbone.
Okay. And for proteins, which are those long chains of amino acids linked by peptide bonds formed through condensation.
Proteolytic enzymes use hydrolysis to add H+ and OH back to those peptide bonds, splitting the protein chain into smaller peptides and ultimately single amino acids.
So the core chemistry powering all this major digestion is this elegant, simple process, hydrolysis, adding water to break bonds.
The only difference then is which specific enzymes are needed for the job.
Exactly. Because each type of bond and molecule needs a precisely shaped enzyme tool to make the reaction happen efficiently.
And all these digestive enzymes are proteins themselves.
Right. That's the first key insight to carry forward. The fundamental chemical strategy is the same break bonds using water, but the biological tools, the enzymes, are highly specialized for each job.
Got it. Simple chemistry, complex tools.
Absolutely. So with that fundamental chemical concept in hand, let's apply it to the specific journey of carbohydrates. When we talk about carbs in our diet, what are the main forms you're actually digesting?
Well, the vast majority of digestible carbohydrates that humans consume come from really just three main sources.
Okay, what are they?
There's sucrose, which is, table sugar. That's a disaccharide.
Right.
Then there's lactose, the disaccharide found in milk.
Milk sugar.
But by far the largest component in most diets is starch. Starch is a large polysaccharide found in non-animal foods like potatoes, grains, legumes, that sort of thing.
Okay, so starch is the big player. The source material also mentioned some minor sources.
Yeah, it did. Things like amylose, glycogen, which is basically animal starch found in meat, small amounts of alcohol, lactic acid, pyruvic acid, pectins, dextrins, and some other minor carbohydrate derivatives you might find in meats.
But these are pretty minor players overall.
Generally insignificant compared to the big three: sucrose, lactose, and especially starch.
And there's one really common carbohydrate that our enzymes just can't handle at all.
Right, that's cellulose. It is a carbohydrate structurally, but the human digestive system simply lacks the specific enzymes needed to hydrolyze the bonds in cellulose.
So it passes through.
It does. While it's important as dietary fiber or roughage, it doesn't provide us with any absorbable nutrients. We just can't break it down.
Got it. So the starch, sucrose, and lactose, they do need breaking down. Where does that start for carbohydrates? Is it the stomach?
Actually, it begins surprisingly early, right in your mouth.
In the mouth.
Yep. When you chew your food, you mix it thoroughly with saliva, and saliva contains an enzyme called chylin.
Chylin.
Chylin is an alpha amylase, secreted primarily by your parotid glands, the big salivary glands.
So it's an amylase, meaning it works on starch. What does it do specifically?
Chylin starts the hydrolysis of starch. It begins breaking those very large starch molecules down into smaller polysaccharides chains of about 3 to 9 glucose molecules, and also into the disaccharide maltose, which is just 2 glucose units linked together.
But, food is only in your mouth for a short time when you chew and swallow. Does that mean very little actually happens there?
Well, it's true that because of the short transit time, typically not more than, say, 5% of the starches are hydrolyzed in the mouth itself.
Okay, only 5%.
However, the digestion of starch by pylin can actually continue in the body and fundus of the stomach for quite a while, maybe up to an hour after you swallow.
How does that work if the stomach is acidic?
Because when you swallow a mouthful of food, it forms a sort of bolus, a mass. This stomach acid penetrates this mass relatively slowly. So the chylin mixed into the center of the food mass can keep working for a while before it gets exposed to the really low pH.
Ah, okay. And that acidity is key then, right?
Yeah.
Because salivary amylase must have an optimal pH.
Exactly. The activity of chylin is completely blocked once the pH of the stomach contents drops below about 4.0 due to mixing with gastric acid.
But it gets some work done before that happens.
Yes, and here's where it gets really interesting and shows some early efficiency. Before the acid completely penetrates the food mass and inactivates the amylase, often as much as 30% to 40% of the starches have already been hydrolyzed to maltose and those other small glucose polymers.
Wow, 30 to 40% just from chewing and that initial period in the stomach.
Yep. So even though it eventually gets shut down by stomach acid, the salivary amylase provides a pretty significant head start in the upper stomach.
That's a substantial contribution before the main digestive event even starts in the small intestine. But the major digestion for carbohydrates, as you said, happens later on.
Oh, absolutely. The small intestine is where the primary work gets done, and a huge part of that is thanks to the pancreas.
The pancreas.
Okay.
It seems to be a real powerhouse for digestive enzymes.
It certainly is for carbohydrates.
Yeah.
Pancreatic secretion contains a very large quantity of pancreatic alpha amylase.
Another alpha amylase. Similar to the salivary one.
Functionally, yes. Almost identical. It also hydrolyzes starch into maltose and those small glucose polymers. But the source material notes that the pancreatic version is several times more powerful.
Ah, okay. So the pancreas brings in the heavy artillery for starch digestion.
Precisely. And its action is incredibly rapid. Within just 15 to 30 minutes after the chyme, that semi-liquid mix of food from the stomach enters the duodenum and mixes with the pancreatic juice.
Yeah.
Virtually all the digestible carbohydrates will have been broken down, mostly into maltose and/or other small glucose polymers.
So pretty much by the time the food leaves the first part of the small intestine, the duodenum and maybe the upper jejunum, the large starches are gone, replaced by mostly disaccharides and short chains.
That's right.
But to be absorbed, they need to be monosaccharides single sugar units.
Yeah.
How does the body get from maltose and short polymers to glucose, fructose, and galactose?
That final step is the job of the epithelial cells that line the small intestine, the enterocytes, specifically the ones covering the villi.
Okay, the cells lining the gut wall.
These cells have a specialized surface facing the intestinal lumen called the brush border. It's made up of thousands of tiny microvilli.
The brush border, right. Very high surface area.
Exactly. And embedded right there in the membranes of these microvilli are 4 crucial enzymes that perform the final act of carbohydrate digestion.
The final polishing crew, right on the surface where absorption is about to happen.
Yeah.
What are these 4 enzymes?
They are lactase, sucrase, maltase, and alpha-dextrinase. These enzymes take the disaccharides and those small glucose polymers that were produced earlier and break them down into their single unit monosaccharides right as they encounter the cell surface.
So lactase takes lactose.
And splits it into 1 molecule of galactose and 1 molecule of glucose.
Makes sense. Sucrase handles sucrose.
Splitting it into 1 molecule of fructose and 1 molecule of glucose.
Okay, and then maltase and alpha-dextrinase work on the maltose and those small glucose polymers that came from starch digestion.
Breaking each of them down into multiple individual glucose molecules. Maltose gives 2 glucose, the polymers give several.
Okay, so the ultimate final products of all carbohydrate digestion are exclusively these monosaccharides: glucose, galactose, and fructose. And that's the form that's ready for absorption into the cells.
Exactly. And this whole sequential process, starting with salivary amylase, hitting hard with pancreatic amylase, and then finishing right at the brush border with these specific enzymes, perfectly explains why glucose is overwhelmingly the most abundant end product.
Because starch is the main dietary carb.
Precisely. Since starch is the dominant dietary carbohydrate for most people, and its breakdown ultimately yields primarily glucose molecules via maltose and polymers, glucose usually represents more than 80% of the final absorbed carbohydrate calories.
And the other 2?
Galactose, from milk lactose, and fructose, from table sugar fruit sucrose, seldom account for more than 10% each of the absorbed monosaccharides.
It's fascinating how the enzymatic machinery is layered throughout the digestive tract. First, the bulk breakdown, then the final snip right at the absorption site, ensuring that by the time we're ready to absorb, the carbs are in their simplest possible units.
It's a very efficient, multi-stage enzymatic breakdown leading to specific single-unit end products ready for uptake. That's a critical takeaway.
Definitely. Okay, let's shift our focus now to proteins, the body's essential building blocks. Structurally, how are proteins built again?
Right. Dietary proteins are complex, long chains made up of many different amino acids. And these amino acids are linked together by what we call peptide bonds.
Peptide bonds, formed by that condensation reaction we talked about.
Exactly. Formed by removing an OH from one amino acid and an H plus from the next, effectively removing a water molecule to create the link. And the specific sequence and type of amino acids in that chain are what determine the unique properties and function of each individual protein.
Okay, and like carbohydrates, does protein digestion also get a head start before the small intestine and the stomach?
Yes, it does, with a very important enzyme called pepsin.
Pepsin, that's the main peptic enzyme in the stomach, right? What are its ideal working conditions?
Pepsin is quite particular about its environment. It's most active in a highly acidic setting, specifically a pH between 2.0 and 3.0.
Very acidic.
Extremely. And it becomes virtually inactive if the pH rises above about 5.0.
And the stomach is responsible for creating that super acidic environment.
Absolutely. Specialized cells in the gastric glands called parietal or oxyntic cells secrete hydrochloric acid, HCl, at an incredibly low pH, somewhere around 0.8.
Wow, 0.8.
Yeah. By the time this acid mixes with the food and other stomach contents, the overall pH usually settles into that ideal 2.0 to 3.0 range, perfect for pepsin activity.
What's a key specific function of pepsin in the stomach? Does it just start chopping proteins randomly?
It does start chopping proteins, but one of its most crucial roles mentioned in the source is digesting collagen.
Collagen, like in connective tissue.
Exactly. Collagen is a major protein found in the connective tissue of meats, and it's quite resistant to breakdown by most other digestive enzymes. Pepsin, however, is particularly good at hydrolyzing collagen fibers.
Why is that so important?
Because digesting the collagen matrix that holds meat together helps to loosen up the meat fibers. This allows other digestive enzymes that come later, further down the tract, to better penetrate the meat and access the other proteins within those fibers.
Ah, so it's like a meat tenderizer enzyme almost.
In a way, yes. The source notes that without pepsin, meat digestion can be significantly impaired because other enzymes can't get in as easily.
So pepsin doesn't completely break down the proteins, but it's essential for starting the process, especially by tackling tough structures like collagen.
Exactly right. Pepsin initiates protein digestion. It typically provides only about 10% to 20% of the total breakdown needed. It works by hydrolyzing certain peptide linkages within the long protein chains.
Chopping them up.
Yeah, converting those long proteins into smaller chains called proteoses, peptones, and some larger polypeptides. It's the initial rough chopping.
And then, similar to the carbohydrate story, the main event for protein digestion shifts downstream to the small intestine.
Correct. The vast majority of protein digestion happens in the upper small intestines, specifically the duodenum and jejunum. And again, this is primarily driven by powerful proteolytic enzymes secreted by the pancreas.
The pancreas, again, bringing the power tools. What are the key pancreatic protein enzymes?
The major ones mentioned are trypsin, chymotrypsin, carboxypolypeptidase, and elastase.
Okay, quite a lineup.
Yeah. As the acidic chyme squirts from the stomach into the duodenum and mixes with the alkaline pancreatic juice, these enzymes rapidly go to work on those partially digested protein fragments, the proteoses, peptones, and polypeptides created by pepsin.
How do these different pancreatic enzymes contribute? Do they all do the same thing?
No, they have different specificities. Trypsin and chymotrypsin are primarily involved in splitting proteins and those larger polypeptides into yet smaller polypeptides. They cut within the chains.
Okay, internal cuts. What about the others?
Carboxypolypeptidase is different. It's an exopeptidase. That means it specifically cleaves individual amino acids off one by one from the carboxyl end, the C-terminus of the polypeptides.
Ah, snipping off the ends.
Right. And elastase, as the name suggests, is important for digesting elastin fibers, which are another component of the connective tissue that helps hold meats together.
Okay, so the pancreatic enzymes are doing a lot more chopping, breaking things down into smaller peptides, and even starting to release some single amino acids. But does the pancreatic juice fully break everything down into individual amino acids right there in the intestinal lumen?
Not completely, no. While they break down the proteins into much smaller units, only a relatively small percentage are fully reduced to single amino acids by the action of pancreatic enzymes alone in the lumen. Most of the protein digestion products at this stage are still in the form of dipeptides, 2 amino acids, and tripeptides, 3 amino acids.
So the final stage of breaking down those dipeptides and tripeptides and getting everything into the absorptive cells happens where?
That final crucial step takes place right at the surface of the small intestinal epithelial cells, the enterocytes, specifically on their brush border microvilli.
Back to the brush border again. It's a busy place.
It really is. These microvilli membranes contain yet another set of enzymes called peptidases that protrude out into the intestinal fluid.
Peptidase is on the brush border. What kinds are important here?
The source mentions 2 main types, aminopolypeptidase and several dipeptidases.
Okay, what do they do?
Aminopolypeptidase works similarly to carboxypolypeptidase, but it splits individual amino acids from the amino end, the N-terminus, of larger polypeptides, further shortening them into tripeptides and dipeptides. The dipeptidases then specifically target and split the remaining dipeptides into 2 single amino acids. They are like the final precise cutting tools right at the point of absorption.
So now we have a mix arriving at the cell membrane. Single amino acids, plus some dipeptides and tripeptides that maybe haven't been fully broken down yet by the brush border enzymes. How do these get inside the enterocyte?
Amino acids, dipeptides, and tripeptides are all readily transported across the microvillar membrane into the interior, the cytosol of the enterocyte.
Okay, they can all get in. And is the job finished once they are inside the cell? Or is there another step?
There's one more crucial step for the peptides. Inside the cytosol of the enterocyte, there are multiple other peptidases.
Intracellular peptidases.
Yes. These cytosolic peptidases are specific for the different types of amino acid linkages that might still exist within those dipeptides and tripeptides that were absorbed intact. Within minutes of entering the cell, these intracellular enzymes digest virtually all the remaining dipeptides and tripeptides into single amino acids.
Wow. So even if they sneak into the cell as small peptides, the cell makes absolutely sure they are broken down into single amino acids before they leave the cell on the other side to enter the bloodstream.
Exactly. It's a critical quality control step.
That's a key insight. Final digestion for proteins occurs not just at the brush border, but also inside the absorbing cell itself.
It ensures that the overwhelming majority, the source says more than 99% of the absorbed protein products that actually enter the bloodstream, are individual amino acids.
And absorbing larger fragments is generally avoided.
Yes, absorption of intact peptides is rare, and absorption of whole protein molecules is extremely rare under normal conditions. When intact whole proteins do manage to get absorbed, it can sometimes trigger allergic or immunological reactions, which really highlights why this thorough breakdown, both outside and inside the cell, is so important.
It makes perfect sense. The body's multi-layered protein digestion system, culminating in that breakdown right on and inside the cell, is incredibly effective at delivering the fundamental amino acid building blocks while minimizing the absorption of larger fragments that could potentially cause problems.
It's a very robust system.
Okay, let's move on to the often trickiest macronutrient to digest and absorb, fats. What are the main types of fats we typically eat?
The most abundant type by far in most diets is neutral fats, or more technically, triglycerides.
Triglycerides, okay.
These are molecules consisting of a glycerol backbone chemically linked to 3 fatty acid chains. Most animal fats and vegetable oils are primarily triglycerides.
Are there other dietary fats we need to consider?
Yes, there are also smaller amounts of phospholipids, which are important components of cell membranes, cholesterol, which is technically a sterol but handled like a fat, and cholesterol esters, cholesterol linked to a fatty acid.
Okay. Do phospholipids and cholesterol esters get digested?
Yes, they contain fatty acids, so they are hydrolyzed by specific enzymes to release those fatty acids, along with the other components. Free cholesterol itself isn't digested in the same way, but its absorption is linked to the fat pathway.
Does any fat digestion happen early on, say, in the stomach?
Only a very small amount, generally considered pretty unimportant for overall fat digestion. There's a small quantity of an enzyme called lingual lipase, secreted by glands in the mouth and swallowed with saliva.
Lingual lipase.
Yeah. It can digest some triglycerides in the acidic environment of the stomach, but it usually accounts for less than 10% of the total fat digestion. It might be slightly more important in infants for digesting milk fat, but in adults, it's minor.
So nearly all significant fat digestion happens where?
In the small intestine, that's the primary site.
Okay. And fats pose a unique challenge compared to carbs and proteins, don't they? They're hydrophobic, they don't mix with water.
Exactly, that's the core problem.
While the digestive enzymes, the lipases, are water-soluble proteins, how does the body allow water-soluble enzymes to effectively attack and digest these water-insoluble fat globules?
That's where the absolutely crucial first step for fat digestion comes in. Emulsification.
Emulsification, breaking big droplets into smaller ones.
Precisely. Think about it. Water-soluble enzymes can only work on the surface of the fat droplets. If fats remain as large globules, the total surface area available for the enzymes to act on would be tiny, making digestion incredibly slow and inefficient.
So the body needs to physically break down those large fat globules into many, much smaller ones.
To dramatically increase the surface area. Yeah.
How does this emulsification happen?
Well, it actually starts with some physical churning and agitation in the stomach, which helps mix the fat into the stomach contents and break it up somewhat. But the vast majority of emulsification, and the really effective part, occurs in the duodenum, the first part of the small intestine.
And what's the key ingredient there for emulsification?
It's bile, which is produced by the liver and stored in the gallbladder, then secreted into the duodenum.
And bile itself doesn't contain digestive enzymes, right?
That's correct. No digestive enzymes in bile. But bile contains a high concentration of 2 crucial types of molecules: bile salts and the phospholipid lecithin.
Bile salts and lecithin.
Both are extremely important for emulsification, with lecithin being particularly effective according to the source.
How do bile salts and lecithin actually achieve this? What's their mechanism?
They essentially act like detergents. Think about dish soap breaking up grease. Bile salt and lecithin molecules have a dual nature. One part of the molecule is very soluble in water, that's the polar or charged part, and another part is highly soluble in fat, the non-polar lipid-soluble part.
Okay, so they have a split personality.
Kind of, yeah. When they encounter a fat globule in the watery chyme, their fat-soluble parts dissolve into the surface layer of the fat droplet. Their water-soluble parts then project outward into the surrounding watery digestive fluid.
What does that arrangement do?
This coating of bile salts and lecithin around the fat droplet significantly lowers the interfacial tension between the fat and the water.
Like making the surface less sticky or resistant to breaking apart?
Exactly. Low interfacial tension makes the fat globules much less stable and much easier to break apart into smaller droplets when agitated.
And there's plenty of agitation in the small intestine.
Oh, yes. The constant churning and mixing movements of the small intestine walls provide the physical force. With the interfacial tension lowered by bile salts and lecithin, this agitation effectively fragments the large fat globules into tiny droplets.
How tiny are we talking?
After effective emulsification and the churning action, the average diameter of the fat particles is reduced to less than 1 micrometer.
That's incredibly small.
It is, and this represents a massive increase in the total surface area exposed to the lipase enzymes. The source estimates as much as 1,000-fold increase compared to the surface area of the unemulsified fat globules.
1,000-fold increase. That makes the detergent function of bile salts and lecithin absolutely vital then. It's the essential prep work for fat digestion.
It truly is. Without effective emulsification, lipase enzymes would have very little surface to work on, and fat digestion would be extremely poor.
Okay, so once the fat is nicely emulsified into these tiny droplets with a huge surface area, then the actual chemical digestion by lipase enzymes can really begin efficiently.
Yes. And here, the main enzyme is pancreatic lipase.
Pancreatic lipase, another key enzyme from the pancreas.
Absolutely the most important one for fat digestion. It's present in the pancreatic secretion in enormous quantities, far more than usually needed to digest a normal meal's worth of fat. And it's incredibly efficient, capable of digesting all the available triglycerides within about a minute once it reaches them on the surface of the emulsified droplets.
Is there any other lipase involved?
There is a small amount of lipase associated with the enterocytes, called enteric lipase. But the pancreatic lipase is by far the dominant player and usually all that's needed.
And what are the main end products when pancreatic lipase digests triglycerides?
The vast majority of triglycerides are split into two main products, free fatty acids and 2-monoglycerides. A 2-monoglyceride is a glycerol molecule with just one fatty acid still attached, specifically at the middle position.
Okay, so the lipase chops off 2 of the 3 fatty acids, leaving 1 attached in the middle, plus 2 free ones. Now, the source mentioned that hydrolysis is a reversible reaction. If these end products, the fatty acids and monoglycerides, just hang around the surface of the fat droplet, couldn't they slow down or even block further digestion?
Yes, that's a potential problem. Product inhibition. If the concentration of free fatty acids and monoglycerides builds up right where the lipase is working, it can indeed slow down the rate of further hydrolysis.
So how does the body deal with that? How does it keep the digestion going efficiently?
This is where bile salts play their second, equally critical function. They remove the monoglycerides and free fatty acids from the vicinity of the digesting fat droplets almost as quickly as they are formed.
How do they remove them?
By forming structures called micelles.
Micelles. Okay, we heard about emulsification. Now micelles, what are they?
When bile salts reach a certain concentration in the chyme, they spontaneously aggregate into tiny water-soluble particles called micelles. These are even smaller than the emulsion droplets, typically about 3 to 6 nanometers in diameter.
Tiny packages.
Very tiny. Each micelle is typically formed by about 20 to 40 bile salt molecules arranging themselves in a specific way.
How are they arranged?
They arrange themselves so their fat-soluble parts cluster inwards, creating a tiny lipid core. Their polar water-soluble parts project outwards, facing the watery chyme.
And the fatty acids and monoglycerides go into that core.
Exactly. The hydrophobic core of the micelle effectively dissolves the fat, digestates the monoglycerides and free fatty acids, pulling them away from the emulsion droplet surface and trapping them inside the micelle.
And because the outside of the micelle is water-soluble.
The whole micelle particle, carrying its fatty cargo, becomes water-soluble and can easily move through the watery chyme.
So they're essentially tiny transport packages or shuttles for the fat breakdown products.
Precisely. They act as a transport medium to carry the relatively water-insoluble monoglycerides and free fatty acids from the emulsion droplets across the unstirred water layer lining the gut wall, right up to the brush borders of the intestinal epithelial cells where absorption happens. They ferry them.
They ferry them to the absorption site.
Okay.
And what happens when these micelles reach the brush border microvilli? Do the micelles get absorbed?
No, this is key. At the brush border, the monoglycerides and fatty acids, being lipid-soluble themselves, readily diffuse out of the micelle core, pass through the lipid bilayer of the epithelial cell membrane, and enter the cell interior.
So the cargo gets dropped off. What happens to the bile salt micelle itself?
Critically, the bile salts themselves generally remain in the chyme. They don't enter the cell with the digestates, they are released back into the lumen.
To go pick up more cargo.
Exactly. To pick up more fat digestates from the emulsion droplets and ferry them to the brush border again. They function like reusable shuttles.
That's incredibly efficient. They don't get consumed in the process. They just facilitate transport and are reused over and over.
Precisely. And this ferrying function of micelles is tremendously important for efficient fat absorption. The source material highlights this starkly. In the presence of plenty of bile micelles, about 97% of dietary fat is normally absorbed.
97%. What happens without them?
Without adequate bile salt micelles, for example, if bile secretion is blocked, fat absorption drops dramatically to only about 40% to 50%.
Wow. That single statistic really underscores the indispensable role of micelles. They aren't the enzymes doing the cutting, but they're absolutely essential for, first, preventing product inhibition during digestion, and second, transporting the water-insoluble end products to the cell surface for uptake.
They are the critical co-players, enabling both efficient digestion and absorption of fats.
What about those other dietary lipids, like cholesterol esters and phospholipids? How are they handled? Does the micelle system work for them too?
Yes, it does. Cholesterol esters and phospholipids also contain fatty acids and are hydrolyzed by other specific lipases present in the pancreatic secretion.
Which enzymes?
There's cholesterol ester hydrolase, which hydrolyzes cholesterol esters into free fatty acids and free cholesterol. And there's phospholipase A2, which hydrolyzes phospholipids, also releasing free fatty acids and other components like lysophospholipids.
And the products?
Just like the monoglycerides and fatty acids from triglycerides, these breakdown products, the free cholesterol, lysophospholipids, and fatty acids, are also incorporated into the bile salt micelles. The micelles then ferry them to the brush border for absorption into the enterocytes.
So the micelle system is the universal transport mechanism for essentially all lipid digestion end products.
Pretty much. The source explicitly states that essentially no cholesterol is absorbed at all without this micelle function. It's that critical for cholesterol uptake.
Amazing. Okay, we've covered the digestion, the breaking down of carbs, proteins, and fats into absorbable units. Now, how do these smaller molecules actually get across that intestinal lining and into the body proper? Before we dive into the specifics for each nutrient, let's set the stage. What are the general principles of absorption and what's special about the gut's anatomy that facilitates it?
Right. To understand absorption across the gut lining, it helps to remember the basic ways substances cross cell membranes. Things like, well, active transport, where the cell uses energy, usually from ATP, to pump substances against a concentration gradient.
Okay, active transport.
Then there's diffusion, where substances simply move passively down their concentration gradient from high concentration to low.
Simple diffusion.
There's also facilitated diffusion, which still involves movement down a gradient, so no direct energy input against the gradient, but it requires the help of a specific carrier protein in the membrane.
Like needing a specific doorway.
Exactly. And finally, there's osmosis, which is specifically the diffusion of water across a semi-permeable membrane, driven by differences in the solute concentration.
Got it. Active transport, diffusion, facilitated diffusion, osmosis. These are the underlying mechanisms we'll see being used in specialized ways in the gut.
Precisely.
And the sheer volume of fluid the intestines have to process and absorb every single day is just mind-boggling when you think about it.
It truly is. On average, you might ingest about, say, 1.5 liters of fluid a day in drinks and food.
Okay.
But your body also secretes about 7 liters of fluid into the gut lumen each day. Saliva, gastric juice, pancreatic juice, bile, intestinal secretions.
7 liters secreted, plus the 1.5 we drink.
That's a total of around 8 to 9 liters entering the small intestine every day that needs to be managed, mostly reabsorbed.
And the small intestine handles the bulk of that reabsorption.
Yes, the healthy small intestine is incredibly efficient. It typically absorbs about 7 to 8 liters of that total daily fluid load.
Leaving how much for the large intestine?
Only about 1.5 liters normally passes through the ileocecal valve into the large intestine each day. And it's important to note, the small intestine's capacity for absorption is far greater than this normal daily load.
It has reserve capacity.
Huge reserve capacity. It could potentially absorb 20 or more liters of water, several kilograms of carbohydrates, and over 500 grams each of protein and fat daily if presented with them.
That's incredible. Interestingly, the source points out that the stomach absorbs very little. Why is that?
Yeah, the stomach is simply not designed for significant absorption. For one, it lacks the specialized absorptive structures, the villi and microvilli that we'll see are crucial in the small intestine.
Okay, no way.
And also, the junctions between the epithelial cells lining the stomach are very tight, which prevents much leakage or passage of substances between the cells. So, the stomach primarily absorbs only small quantities of highly lipid-soluble substances that can pass directly through the cell membranes easily.
Like what?
Like alcohol and certain drugs like aspirin, but not significant amounts of water, ions, or nutrients.
So the small intestine is the undisputed champion of absorption, and its incredible power comes largely from its structure, which creates an absolutely massive surface area for absorption.
This anatomical design for maximizing surface area is truly ingenious. The absorptive surface isn't just the simple inner lining of a smooth tube, it's amplified dramatically at multiple levels.
Okay, what's the first level of amplification?
First, you have the large folds of the lining called the folds of Kerckring, or valvulae conniventes. These are large, shelf-like circular folds of the mucosa and submucosa that protrude significantly into the lumen, sometimes up to 8 millimeters.
And they are throughout the small intestine.
They are especially prominent and numerous in the duodenum and jejunum, the upper parts where most absorption occurs. These folds alone increase the total absorptive surface area about threefold compared to a smooth cylinder.
3 times just from the large folds. Then, sitting on top of those folds are the villi.
Yeah, right. Exactly. Millions of them. These are tiny finger-like or sometimes leaf-like projections of the mucosa itself, typically about 1 millimeter long. They cover the entire surface of the small intestine's folds.
Like a velvety texture.
Precisely. They are densest in the upper small intestine and become a bit sparser further down. The presence of these countless villi increases the available surface area about another tenfold.
Okay, so 3x from folds times 10x from villi. It's 30x already. And there's a third level.
Yes, the third level of amplification is the microvilli. These form the brush border on the surface of each individual epithelial cell that covers the villi.
On every single cell.
Yes, each absorptive enterocyte has as many as 1,000 tiny microvilli projecting from its luminal surface. Each microvillus is only about 1 micrometer long, but collectively they form that dense brush border facing the chyme.
And how much do the microvilli increase the area?
This adds yet another massive layer of surface area amplification, increasing it by at least 20-fold again.
Wow. So let's multiply those factors. About 3x from the folds times 10x from the villi times 20x for the microvilli. That gives a combined increase of, 600 times the surface area of just a smooth tube of the same length.
Exactly. And the source actually states the total increase is nearly 1,000-fold, suggesting the effective overall amplification might be even higher.
1,000-fold increase. What does that translate to in actual area?
It results in an astonishing total absorptive surface area for the human small intestine at 250 square meters or more.
250 square meters. That's enormous. How big is that relatively?
To put that into perspective, that's roughly the size of a singles tennis court, all packed efficiently inside your abdomen.
A tennis court of absorptive surface lining your gut. That scale is just incredible. It really highlights the body's absolute commitment to maximizing nutrient uptake.
It's truly remarkable engineering, and the internal structure of each individual villus is also perfectly designed for transporting the absorbed material away efficiently.
What's inside a villus?
Inside each villus, there's a rich network of blood capillaries. An arteriole enters the base, branches into capillaries that run just under the epithelium, and then these capillaries converge into venules that exit the villus. This vascular system readily collects absorbed water and most dissolved nutrients like sugars and amino acids, carrying them into the portal blood circulation, which heads straight to the liver for processing.
But there's also a separate system inside the villus for fats, isn't there?
Yes. Crucially, each villus also contains a central lymphatic vessel called a lacteal. This lymphatic system is the primary route for absorbing most dietary fats after they are processed inside the enterocyte, as we'll detail shortly.
Okay. Blood for water, sugars, amino acids, lymph for fats. The source also briefly mentions penocytosis as an absorption mechanism.
Yes. Penocytosis is a process where the cell membrane can invaginate and pinch off tiny vesicles containing small amounts of fluid and dissolved substances from the lumen, effectively engulfing them.
Is it important for nutrient absorption?
While it might play a minor role in absorbing very small quantities of certain substances, including perhaps some large molecules like intact proteins in newborns, it's generally not considered the main mechanism for the bulk absorption of water, ions, or the major macronutrient breakdown products in adults. Active transport and diffusion are much more significant.
And one more detail about the brush border. It isn't just static, right? It moves.
That's right. The microvilli contain tiny contractile filaments made of actin. These filaments cause rhythmic contractions and movements of the individual microvilli.
What does that movement achieve?
It keeps the microvilli constantly agitated and swirling within the layer of chyme right next to the cell surface. This constant movement helps to continuously expose the absorptive surfaces to fresh quantities of chyme, preventing stagnation and further aiding the efficiency of absorption by optimizing contact.
So the anatomy is not just about maximizing static area. It's also about dynamically optimizing contact with the food mixture. That makes sense. The combined effect of folds, villi, and microvilli, creating that tennis court-sized surface, along with the efficient vascular and lymphatic drainage and the dynamic brush border movement. It truly makes the small intestine an unparalleled biological machine for absorption.
Couldn't agree more.
Okay, let's delve into the specifics now. How exactly are water and those crucial electrolytes absorbed? Water, as you mentioned, is handled in massive volumes. How is it absorbed? Is it pumped?
No, water absorption in the intestine is driven entirely by diffusion. It strictly follows osmotic gradients. There are no active pumps for water itself in the gut lining.
So water just moves passively based on concentration differences of solutes.
Exactly. When the chyme in the intestinal lumen becomes sufficiently dilute compared to the fluid inside and around the epithelial cells and in the blood, meaning the chyme has a higher water concentration, water is absorbed through the intestinal mucosa into the blood of the villi simply by osmosis. It moves from the area of higher water concentration, the dilute chyme, to the area of lower water concentration, the plasma and intercellular fluid.
What if the chyme is more concentrated in the blood, like after drinking a very sugary drink?
Then the osmotic gradient is reversed. Initially, water can actually move from the plasma into the chyme in the duodenum to try and dilute the hyperosmotic solution, bringing it closer to being isosmotic, having the same osmotic pressure with the body fluids. Once it's isosmotic or hypotonic, then net water absorption into the blood can occur further down.
Okay. And can water move through the cells themselves, or does it go between them?
It actually moves both ways. Water can pass directly through the epithelial cells, this is called the transcellular pathway, and also squeeze between the cells through the relatively permeable tight junctions that link them together. This is the paracellular pathway.
Which pathway is more important?
Both are significant. Importantly, the movement of ions, especially sodium, into the narrow paracellular space between the cells is key. This builds up a high osmotic concentration in that space.
Creating a local osmotic gradient.
Exactly. And that high concentration in the paracellular space pulls water by osmosis both through the cells, transcellularly, and between the cells, paracellularly, into that space, and from there it flows readily into the blood capillaries within the villus.
So sodium movement is tightly linked to water movement. Speaking of sodium, it's a huge player in electrolyte balance. How much sodium are we talking about cycling through the gut, and why is its absorption so absolutely critical?
Sodium is incredibly vital, and the numbers are staggering. Your body secretes about 20 to 30 grams of sodium into the intestine every single day, primarily in digestive juices. On top of that, the average person eats maybe 5 to 8 grams of sodium daily.
So altogether, 25 to 35 grams of sodium enters the gut each day.
Roughly, yes. To maintain sodium balance, the intestines have to absorb almost all of that back 25 to 35 grams daily. That's a significant portion of your body's total sodium content cycling through the gut lumen and back into the body each and every day.
And the body is normally incredibly efficient at reclaiming it, right?
Under normal conditions, fantastically efficient. Usually, less than 0.5% of the total intestinal sodium load is actually lost in the feces. Sodium is absorbed so rapidly and completely along the length of the intestine that virtually none is left to be excreted by the end.
But this high efficiency also means that if something goes wrong,
Exactly. It's why severe diarrhea can be so dangerous so quickly. If those massive intestinal secretions, which are rich in sodium, are lost to the exterior instead of being reabsorbed, the body can rapidly become critically depleted of sodium. This can lead to circulatory collapse and potentially lethal outcomes within just hours if not treated promptly.
So preventing sodium loss is job one. But sodium also has a crucial dual role beyond just maintaining balance. You mentioned it helps absorb other things.
Yes, this is a critically important point. Sodium ions are key co-players in the absorption of many other essential nutrients, specifically most sugars like glucose and galactose, and almost all amino acids.
How does that work?
It happens through mechanisms called sodium cotransport or secondary active transport.
Okay, let's break down the core mechanism of sodium absorption itself first, because that seems to power these other processes. How does sodium get absorbed from the gut lumen into the blood?
The primary engine driving sodium absorption across the intestinal epithelium is an active transport pump located on the basal and lateral cell membranes of the epithelial cells. That's the side facing away from the gut lumen and towards the underlying tissue and blood vessels.
Okay, on the back side of the cell.
Right. This pump is the famous sodium-potassium ATPase pump, Na plus K plus ATPase. It actively pumps sodium ions out of the epithelial cell cytoplasm and into the narrow paracellular space between the cells.
Pumping sodium out, does it pump anything in?
Yes, it simultaneously pumps potassium ions into the cell, but the key effect for sodium absorption is pumping sodium out. This process requires a significant amount of cellular energy in the form of ATP.
And what does actively pumping sodium out of the cell achieve inside the cell?
It achieves a drastically low intracellular sodium concentration. The sodium level inside the enterocyte is kept very low, typically around 50 milliequivalents per liter, MEQL.
And how does that compare to the sodium level in the gut lumen?
The sodium concentration in the intestinal chyme is typically much higher, usually around 142 MEQL, which is similar to the concentration in blood plasma.
So pumping sodium out the back creates this massive electrochemical gradient for sodium across the brush border membrane, the side facing the lumen.
Exactly. There's a much lower concentration of sodium inside the cell than outside in the lumen, and there's also an electrical gradient. The inside of the cell is typically negatively charged relative to the lumen. Both factors strongly favor the movement of positively charged sodium ions into the cell from the lumen.
So sodium wants to rush into the cell down this gradient?
Precisely. And this steep downhill gradient provides the driving force for sodium entry across the brush border membrane through specific sodium channels and transporters located there.
And this downhill movement of sodium is coupled with the transport of other substances, right? This is the co-transport or secondary active transport you mentioned earlier.
Yes, this is where the brilliance of the system really shines. In addition to simply moving into the cell via channels, sodium ions also bind to various specific carrier proteins embedded in the brush border membrane. The source highlights SGLT1, which transports sodium along with glucose or galactose.
SGLT1 for sugars.
Several different sodium amino acid co-transporters.
Or protein.
And also a sodium hydrogen exchanger, which brings sodium in while pumping hydrogen ions out. Now, these carrier proteins typically require both the sodium ion and the other specific substance, like glucose, an amino acid, or maybe another ion, to bind to them simultaneously before they'll transport either one across the membrane.
Both have to be there.
Usually, yes.
Yep.
And when the sodium ion moves down its steep electrochemical gradient into the cell through one of these cotransporters, it literally drags the other substance along with it.
Even if that other substance is moving against its own concentration gradient.
Yes, that's the key. The energy stored in the sodium gradient is used to pull the glucose or amino acid into the cell, even if the concentration of glucose or amino acid is already higher inside the cell than in the lumen. The energy for moving the glucose or amino acid uphill is supplied indirectly by the sodium-potassium ATPase pump on the basolateral membrane, because that pump is what created the sodium gradient in the first place.
Wow. So the active pumping of sodium out the backside of the cell sets up the crucial condition, the low intracellular sodium, that then powers the entry of sodium and these other essential nutrients through the front door, the brush border. That's a fundamental insight into how a huge amount of nutrient absorption works.
It's a highly efficient and clever system, leveraging the primary active transport of sodium to drive the secondary active transport of many other vital molecules.
And it's this buildup of sodium and chloride, which tends to follow sodium electrically in that narrow paracellular space after being pumped out by basolateral pumps, that creates the osmotic gradient we talked about earlier for water absorption.
Precisely. That high concentration of ions, primarily sodium chloride, accumulating in the paracellular space, makes that space hyperosmotic compared to the lumen and even the cell interior.
And that osmotic difference pulls the water along.
Exactly. Water then moves by osmosis from the lumen, both through the cells, transcellularly, and between the cells via the tight junctions, paracellularly, into that hyperosmotic paracellular space, and from there it flows easily into the blood capillaries. So water absorption is intricately linked to and largely powered by active sodium transport.
Does anything regulate sodium absorption, especially if the body is, say, dehydrated and needs to conserve salt and water?
Yes, a key hormone involved is aldosterone.
Aldosterone from the adrenal glands.
Yes, secreted by the adrenal cortices. When you become dehydrated or depleted of sodium, your body releases more aldosterone. Aldosterone then acts on the intestinal epithelial cells, particularly in the colon.
What does it do there?
Within about 1 to 3 hours, aldosterone significantly enhances the activity of the enzyme systems and transport mechanisms involved in sodium absorption by the intestinal epithelium. It basically tells the cells to absorb sodium more aggressively.
And if the gut absorbs more sodium, it would then lead to more water being absorbed too.
Exactly. The increased sodium absorption driven by aldosterone causes a secondary increase in the absorption of chloride ions, which follow the sodium, and critically, water, which follows the salt. This effect is especially important in the colon, where it helps the body prevent virtually any significant loss of sodium chloride and minimize water loss in the feces during times of need.
It's the same vital conservation mechanism that aldosterone employs in the kidney tubules, right?
Precisely the same hormonal control system working in both the gut and the kidneys to maintain fluid and electrolyte balance.
Okay, that covers sodium and water. How about chloride absorption? Does it just follow sodium passively?
In the upper parts of the small intestine, duodenum and jejunum, chloride absorption is indeed very rapid and occurs mainly by passive diffusion. It essentially follows the positively charged sodium ions as they are actively absorbed, moving down the electrical gradient created by sodium transport.
So electrical attraction pulls chloride along in the upper gut. Is it different further down?
Yes, in the lower part of the ileum, and especially in the large intestine, chloride is also absorbed via a different, more active mechanism. There's a specific brush border exchanger protein that absorbs a chloride ion from the lumen while simultaneously secreting a bicarbonate ion, HCO3, into the lumen. Chloride then exits the cell into the blood via specific chloride channels on the basolateral side.
A chloride bicarbonate exchange.
Yeah.
Okay. Now, what about bicarbonate itself? You mentioned it's secreted in large amounts in pancreatic juice and bile, so the body needs to reclaim most of it, right? How is bicarbonate absorbed, especially in the upper small intestine where it's neutralizing stomach acid?
Right. Large quantities of bicarbonate are secreted into the duodenum and jejunum, and most of this definitely needs to be reabsorbed to maintain acid-base balance. This happens via an interesting indirect mechanism that's coupled with sodium absorption.
How does that work?
As sodium ions are absorbed into the epithelial cells, moderate amounts of hydrogen ions, H+, are secreted out into the lumen in exchange for some of the sodium, using that sodium-hydrogen exchanger we mentioned.
Okay, H plus goes out, and A plus comes in.
These secreted hydrogen ions then immediately combine with the bicarbonate ions, HCO3, present in the lumen to form carbonic acid, H2CO3.
H plus HCO3 gives H2CO3.
Right, and carbonic acid is unstable. It quickly dissociates into water, H2O, and carbon dioxide, CO2, catalyzed by an enzyme called carbonic anhydrase present in the brush border.
So the bicarbonate effectively becomes CO2 and water.
Exactly, and carbon dioxide is highly lipid soluble, so it rapidly diffuses across the epithelial cell membrane into the cell and then into the blood. The blood carries it to the lungs where it's eventually exhaled. So even though bicarbonate ions themselves aren't directly transported back in large amounts, this process effectively reclaims the bicarbonate by converting it to absorbable CO2. The source calls this active absorption of bicarbonate, and it's a vital mechanism for neutralizing stomach acid efficiently and reclaiming that essential alkaline bicarbonate buffer. It's the same fundamental mechanism used in the kidney tubules to reabsorb bicarbonate from the filtrate.
That's clever, converting it to gas to get it back.
Yeah.
But you also mentioned bicarbonate secretion in the ileum and large intestine earlier, using that chloride bicarbonate exchanger. Why secrete it there?
Yes, that's the other side of the coin, happening further down the tract. The epithelial cells in the distal ileum and the large intestine have a special capability to actively secrete bicarbonate ions into the lumen in exchange for absorbing chloride ions.
What's the purpose of secreting bicarbonate there?
This bicarbonate secretion serves an important purpose. It helps to neutralize the acidic end products that are constantly being produced by the massive population of bacteria residing and fermenting undigested material in the large intestine. It helps protect the colonic mucosa from acid damage.
So absorption in the upper gut, secretion in the lower gut, both linked to other ion movements. It's a very complex system of absorption and secretion, all tightly regulated for balance. What happens when something goes terribly wrong with this balance, like in severe diarrhea caused by infections?
That's exactly where you see this delicate balance thrown completely out of whack, leading to potentially catastrophic fluid and electrolyte loss. Normally, the crypts of Lieberkühn, which are the pits between the intestinal villi, contain immature epithelial cells that actively secrete some sodium chloride and water into the lumen. This secretion provides a fluid vehicle to help flush nutrients towards the absorptive villi.
So some secretion is normal.
Yes, a basal level of secretion is normal and necessary. The mature absorptive cells on the villi then normally reabsorb this fluid, along with the ingested water and electrolytes. But certain bacterial toxins, most famously the toxin produced by Vibrio cholerae, the bacterium that causes cholera, can stimulate this crypt secretion pathway dramatically and uncontrollably.
How does something like cholera toxin cause such massive secretion? What's the mechanism?
The source explains it quite clearly. A subunit of the cholera toxin gets inside the crypt epithelial cells. Once inside, it persistently activates an enzyme called adenyl cyclase, leading to excessive formation of an intracellular signaling molecule called cyclic adenosine monophosphate, or cyclic AMP.
Okay, too much cyclic AMP. What does that do?
This excess cyclic AMP then has a major effect. It causes the opening of a huge number of chloride channels, specifically the CFTR channel, located on the luminal apical membrane of the crypt cells. This results in a massive continuous efflux of chloride ions out of the cells and into the crypt lumen.
So chloride pours out into the gut.
Yes. This large movement of negative charge out of the cell is thought to then activate sodium pumps or canals, causing sodium ions to follow the chloride out into the lumen as well to maintain electrical neutrality. The net result is a large accumulation of sodium chloride in the crypt lumen.
And salt draws water.
Precisely. This high concentration of NaCl in the lumen creates a powerful osmotic gradient that pulls massive amounts of water out of the blood, across the crypt epithelium, and into the intestine lumen via osmosis.
So the toxin essentially hijacks the cell's normal signaling machinery and forces it to pump out enormous quantities of salt and water uncontrollably.
Exactly. The rate of crypt secretion stimulated by the toxin can be absolutely enormous, producing 5 to 10 or sometimes even more liters of isotonic fluid per day. This secretory volume completely overwhelms the absorptive capacity of the villi cells and the large intestine further down,
leading to that characteristic severe watery diarrhea.
Yes, resulting in rapid, severe dehydration and electrolyte loss, especially sodium, chloride, bicarbonate, and potassium, which can be lethal within hours if not treated urgently. Treatment involves aggressively replacing the lost fluid and electrolytes, typically with oral rehydration solutions containing glucose and salts, or intravenously if needed. The source notes that this massive flushing action might theoretically help wash away the bacteria, but the cost to the body's fluid and electrolyte balance is absolutely devastating.
That story of the cholera toxin really drives home just how critical and finely balanced the normal intestinal absorption of water and electrolytes is for our survival. It's a powerful system, but vulnerable to disruption.
Absolutely.
What about other important ions that we need, like calcium, iron, potassium, magnesium, and phosphate? How are they absorbed?
These are also actively absorbed by the intestinal epithelium, but generally in much smaller quantities compared to sodium, potassium, or chloride, reflecting the body's lower daily requirements for them.
Let's take calcium. How's it handled?
Calcium ions are actively absorbed, primarily in the duodenum. This absorption process is very tightly regulated based on the body's needs for calcium, for bones, muscle function, etc. It's significantly controlled by two key factors: parathyroid hormone, PTH, and vitamin D.
How do they interact?
Parathyroid hormone, released when blood calcium is low, stimulates the activation of vitamin D, specifically conversion to calcitriol in the kidneys. This activated vitamin D is then crucial because it travels to the intestinal epithelial cells and significantly enhances their ability to absorb calcium from the gut lumen by increasing the expression of calcium channels and binding proteins.
So vitamin D is essential for getting calcium out of our food.
Absolutely. Without sufficient activated vitamin D, calcium absorption is very poor, even if dietary intake is adequate.
And iron absorption, also regulated.
Yes, iron ions are also actively absorbed from the small intestine, mainly in the duodenum as well. The rate of iron absorption is very carefully regulated based on the body's current iron stores and its need for iron, especially for the formation of hemoglobin in red blood cells. When body stores are low, absorption increases. When stores are high, absorption decreases to prevent iron overload, which can be toxic.
You mentioned earlier that these bivalent ions like calcium and iron, which have a +2 charge, are absorbed in much smaller quantities than monovalent ions like sodium +1 or chloride -1.
Yes, that's a general principle highlighted in the source. Monovalent ions like sodium, potassium, and chloride are absorbed with relative ease and in very large quantities along the intestine. Bivalent ions like calcium, magnesium, phosphate, which is polyatomic but effectively multivalent, and iron are typically absorbed in only small amounts, even though their transport is active.
Can you give a sense of the scale difference?
The source gives a striking comparison. The maximum rate at which calcium ions can be absorbed is only about 150th of the normal rate at which sodium ions are absorbed.
Wow. 50 times less capacity for calcium than for sodium.
Exactly. Fortunately, the body only requires relatively small quantities of these bivalent ions daily compared to sodium, so the active transport mechanisms, although lower capacity, are generally sufficient to meet our physiological needs, provided dietary intake and regulatory factors, like vitamin D for calcium, are adequate.
So even though the scale of transport is smaller, the active regulated absorption ensures the body gets the essential bivalent minerals it needs for critical functions like bone health, oxygen transport, enzyme activity, and so on.
That's the idea, precision over bulk.
Okay, we've looked at how water and all these different ions get absorbed. Now let's get to the core reason we eat.
Yeah.
Absorbing the energy-providing nutrients that break down products of carbs, proteins, and fats into the bloodstream so our cells can use them.
Right. We've already established how these large macronutrients are broken down into their smaller absorbable forms, primarily monosaccharides, glucose, galactose, fructose from carbohydrates, mostly dipeptides, tripeptides, and some free amino acids from proteins, and mainly monoglycerides and free fatty acids from fats, plus cholesterol.
Let's start with the monosaccharides from carbohydrate digestion. How are they absorbed across the brush border?
As we discussed, carbohydrates are absorbed almost entirely as monosaccharides. Very little absorption of disaccharides occurs, and almost none of larger molecules. Glucose is the most abundant, making up over 80%.
And how do glucose and its close relative, galactose, get into the enterocyte?
Glucose and galactose are both absorbed using a sodium co-transport mechanism. It's that same secondary active transport system we discussed in detail for sodium itself.
Can you quickly recap it specifically for glucose?
Sure. The sodium-potassium ATPase pump on the basolateral membrane works constantly to keep the intracellular sodium concentration very low. On the brush border membrane, facing the lumen, there's a specific carrier protein called SGLT1, sodium glucose linked transporter 1.
SGL.
This SGLT1 protein has binding sites for both a sodium ion and a glucose molecule, or a galactose molecule that transports both. Both substances must bind for the transporter to undergo the conformational change that moves them across the membrane. Because intracellular sodium is kept so low, sodium naturally wants to move down its steep electrochemical gradient into the cell via SGLT1.
And it drags the sugar along with it.
Exactly. It effectively pulls the glucose or galactose molecule into the cell along with it, even if the concentration of sugar inside the cell might already be higher than in the lumen. The energy stored in the sodium gradient fuels the uphill movement of the sugar.
So the energy spent pumping sodium out the back ultimately fuels the uptake of glucose and galactose through the front.
Okay.
And once the glucose or galactose is inside the cell cytoplasm, how does it get out the other side to enter the bloodstream?
It then needs to exit the cell across the basolateral membrane. This exit is accomplished by facilitated diffusion using a different transport protein called GLUT2.
GLUT2, facilitated diffusion, so it just moves down its concentration gradient.
Yes, the concentration of glucose and galactose builds up inside the cell due to the active uptake by SGLT1. GLUT2, located in the basolateral membrane, simply provides a pathway for these sugars to move down their concentration gradient out of the cell, into the paracellular space, and then quickly into the capillaries of the villus.
Okay, so for glucose and galactose, active sodium cotransport, SGLT1, on the lumen side for entry, followed by passive facilitated diffusion, GLUT2, on the blood side for exit. What of the other main monosaccharide, fructose? You mentioned its absorption mechanism is different.
Yes, that's important. Fructose does not use the sodium cotransport mechanism. It is absorbed entirely by facilitated diffusion across both the brush border and basolateral membranes.
No sodium involved for fructose. What transporters does it use then?
On the luminal side of the brush border, fructose enters the enterocyte via a specific facilitated diffusion transporter called GLUT5.
GLUT5 for fructose entry.
Then, on the basolateral side, fructose exits the cell into the blood via the same GLUT2 transporter that's also used by glucose and galactose.
So GLUT2 handles exit for all three monosaccharides. But the entry mechanism is different for fructose, GLUT5, compared to glucose-galactose, SGLT1. Does this difference in entry mechanism affect the overall rate of fructose absorption?
Absolutely. Because fructose transport into the cell relies only on facilitated diffusion, GLUT5, and isn't actively coupled to the powerful sodium gradient, its overall rate of transport through the intestinal epithelium is significantly slower, only about half that of glucose or galactose absorption.
Ah, that makes sense. The active pumping for glucose-galactose makes their uptake faster and more efficient.
Right. The source also adds an interesting detail. Apparently, some of the fructose that enters the epithelial cell via GLUT5 can be phosphorylated inside the cell and then enzymatically converted into glucose. This newly formed glucose can then exit the cell via GLUT along with the directly absorbed glucose.
So the cell can even convert some fructose to glucose before sending it into the blood. Interesting. But the bottom line is, glucose and galactose have a more robust, actively driven uptake system compared to fructose's reliance on slower facilitated diffusion. This helps explain again why glucose is the primary energy currency absorbed after a typical mixed meal containing starch.
Precisely.
Now let's move to protein absorption. You established that the final digestion steps inside the enterocyte mean that mostly single amino acids enter the blood, but what forms are actually absorbed across the brush border into the cell? You mentioned dipeptides, tripeptides, and some amino acids.
Correct. After the action of pancreatic enzymes in the lumen and peptidases on the brush border, a mixture of free amino acids, dipeptides, and tripeptides are presented to the enterocyte surface for absorption.
And how do these get into the cell? Does sodium play a role here too?
Yes, for the vast majority of them, energy is supplied primarily by sodium cotransport, operating in a very similar way to how glucose is absorbed.
So sodium is key for absorbing protein breakdown products as well.
Definitely. There are specific transport proteins embedded in the microvilli membrane that have binding sites for a specific amino acid or a small peptide, or tripeptide. These transporters also require the simultaneous binding of a sodium ion.
Both need to bind again.
Yes. Then, as the sodium ion moves down its steep electrochemical gradient into the cell, it pulls the amino acid or peptide along with it, again utilizing the energy stored in the sodium gradient, classic secondary active transport.
Given the variety of amino acids, 20 different types, and peptides, are there many different types of these sodium-dependent transporters?
Yes, quite a few. Because of the diverse chemical properties, size, charge, side chains of the different amino acids and small peptides, the intestinal epithelial cells express multiple different types of sodium cotransporters, each specific for certain classes of amino acids or peptides. The source mentions there are at least 10 different types known.
Are there any exceptions? Do any amino acids get absorbed without sodium?
Yes, the source mentions that a few amino acids can also be transported by facilitated diffusion via special membrane proteins, similar to how fructose is absorbed. This pathway doesn't require sodium coupling, but it's thought to be less significant quantitatively than the sodium cotransport mechanisms for most amino acids.
So sodium cotransport is clearly the dominant mechanism for absorbing the breakdown products of both carbohydrates, glucose, galactose, and proteins, amino acids, small peptides, powerfully leveraging that sodium gradient created by the basolateral Na plus K plus pump.
It's a unifying principle for much of nutrient absorption.
And just to absolutely nail it down, once those dipeptides and tripeptides get inside the cell via these transporters,
they are very rapidly and completely digested into individual amino acids by those multiple cytosolic peptidases residing within the enterocyte cytoplasm. So almost all of the protein products that ultimately exit the cell across the basolateral membrane and enter the portal blood are single amino acids.
Perfect. Okay, so sodium cotransport covers sugars and proteins. Finally, how are fats absorbed? We know they are broken down into monoglycerides and fatty acids and then ferried to the brush border by micelles. What happens right at the cell surface?
Right. After pancreatic lipase does its work, the resulting monoglycerides and free fatty acids, along with cholesterol and other lipid digestates, become dissolved in the hydrophobic cores of the bile salt micelles. These micelles then ferry their lipid cargo through the unstirred water layer to make contact with the brush border surface of the microvilli.
And at the brush border membrane, how do the lipids get across?
At the brush border, the monoglycerides and free fatty acids, being lipids themselves, are readily soluble in the lipid bilayer of the epithelial cell membrane. So they simply diffuse out of the micelle core and directly across the cell membrane into the interior of the epithelial cell.
Passive diffusion. No transporters needed for the lipids themselves.
Generally, yes, simple diffusion across the lipid membrane is considered the primary mechanism for fatty acid and monoglyceride uptake. The key distinction here is that the bile salts that form the micelle do not enter the cell. They release their cargo at the membrane surface and remain in the chyme.
They stay behind to be reused for more ferrying.
Exactly. They shuttle back, pick up more lipids, and ferry them to the surface again. This reuse is, as we've stressed, absolutely vital for achieving that high 97% efficiency of fat absorption. Without the micelles constantly delivering lipids to the surface, diffusion across the membrane would be much, much slower.
Okay, so the monoglycerides and fatty acids diffuse into the enterocyte. What happens to them inside the cell? Do they just pass through to the blood?
No, this is where fat absorption takes a very different turn compared to carbs and proteins. Once inside the enterocyte cytoplasm, the absorbed monoglycerides and free fatty acids are quickly taken up by the cell's smooth endoplasmic reticulum, smooth ER.
The smooth ER. And what happens there? Something unique.
Yes. Inside the smooth ER, the absorbed monoglycerides and fatty acids are enzymatically reesterified. They're reformed into new triglyceride molecules.
Wait, what? The body just spent energy breaking down triglycerides into monoglycerides and fatty acids, absorbs those pieces, and then immediately uses energy to rebuild them back into triglycerides inside the absorbing cell. That seems incredibly counterintuitive. Why do that?
It does seem strange at first glance, but it's a crucial part of the unique packaging and transport pathway the body uses for most absorbed fats. It prevents fatty acids from building up freely in the cell, which could be toxic, and prepares them for export.
Okay, so triglycerides are reformed in the smooth ER. What happens next? How are these newly synthesized triglycerides packaged for export?
These new triglycerides, along with absorbed cholesterol, which also gets reesterified, and phospholipids, aggregate within the ER and are then coated with a layer of specific proteins, called apolipoproteins, synthesized by the enterocyte. This process forms relatively large lipoprotein particles called chylomicrons.
Chylomicrons. Okay, so they are little packages of reassembled fat and cholesterol wrapped in protein.
Essentially, yes. Think of them as tiny transport vehicles specifically designed to carry large amounts of dietary fat through the body's aqueous environment.
How do these chylomicrons get out of the enterocyte and into the circulation? Do they go into the blood capillaries like sugars and amino acids?
No, and this is another major difference. The chylomicrons are released from the basal side of the epithelial cells by exocytosis. However, these chylomicron particles are relatively large, typically 80 to 500 nanometers in diameter. They are much, much larger than single amino acids or monosaccharides.
Too big for the blood capillaries.
Exactly. They are too large to pass directly through the pores or junctions of the small blood capillaries located within the core of the villus. Instead, they enter the much more permeable, large diameter central lacteal, the lymphatic vessel running up the center of each villus.
They go into the lymph system, not the blood directly.
Correct. From the central lacteal, the lymph fluid, now milky with chylomicrons, it's called chyle, flows up through the larger lymphatic vessels of the mesentery, eventually collecting in the major lymphatic vessel called the thoracic duct.
And where does the thoracic duct lead?
The thoracic duct runs up through the chest and finally empties its contents, the lymph carrying the chylomicrons full of dietary fat directly into the large veins of the bloodstream, typically near the junction of the subclavian and internal jugular veins near the heart.
So most absorbed dietary fat bypasses the portal vein system and the liver initially. It takes this indirect route via the lymphatic system to enter the general circulation. That's a really major difference compared to how carbohydrates and proteins are handled.
It absolutely is. Remember, carbohydrates and proteins, absorbed as monosaccharides and amino acids, go directly into the capillary blood of the villi. This blood collects in the portal vein, which carries these nutrients straight to the liver for initial processing before they enter the general circulation. Most dietary fats, packaged as chylomicrons, go via the lymph, effectively bypassing the liver on their first pass through the circulation.
Is there any exception to this lymphatic route for absorbed fats? Do any fats go directly into the blood?
Yes, there is an important exception for short chain and medium chain fatty acids. These are typically fatty acids with fewer than about 12 or 14 carbon atoms in their chain. Examples are found in butterfat and coconut oil.
What's different about them?
These shorter chain fatty acids are significantly more water soluble than the long chain fatty acids that make up most dietary fat. Because of this, they're mostly not reconverted into triglycerides within the smooth ER of the enterocyte.
So they stay as free fatty acids inside the cell.
Yes. And because they remain as relatively small, somewhat more water-soluble free fatty acids, they can actually diffuse directly from the intestinal epithelial cells across the basolateral membrane and into the capillary blood within the villi, just like glucose and amino acids do.
So they do enter the portal circulation and go to the liver first.
Correct. They bypass the whole chylomicron formation and lymphatic transport pathway used by long-chain fatty acids.
So the length of the fatty acid chain determines its absorption pathway after it enters the cell. Shorter ones go to blood, longer ones get repackaged and go to lymph. That's another layer of remarkable metabolic specialization.
It really highlights how the body adapts its handling strategies based on the chemical properties of the nutrients.
Incredible. Okay, we've journeyed through the small intestine's amazing digestion and absorption processes for all the macronutrients, plus water and ions. But the journey isn't quite over yet, is it? The remaining contents pass from the ileum into the large intestine, which has its own crucial role to play, particularly in absorption.
Exactly right. As we mentioned, around 1,500 milliliters or 1.5 liters of chyme, still containing a significant amount of water and electrolytes that weren't absorbed in the small intestine, normally enters the large intestine each day through the ileocecal valve.
And the large intestine's main absorptive job is?
Its primary function in terms of absorption is to soak up most of that remaining water and electrolytes, effectively concentrating the undigestible waste material into feces.
And it's pretty effective at this final reclamation step.
Remarkably effective under normal conditions. The large intestine efficiently reduces that incoming 1,500 milliliters of fluid down to less than 100 milliliters of water that is typically lost in the feces each day. It also avidly absorbs virtually all the remaining sodium and chloride ions, leaving only about 1 to 5 milliequivalents, MEQ, of each to be excreted.
Is the entire length of the large intestine equally involved in this absorption?
Not quite. The source material specifies that most of this water and electrolyte absorption occurs in the proximal half of the colon, the ascending and transverse colon. This region is often functionally referred to as the absorbing colon.
And the distal half? The descending colon, sigmoid colon, rectum.
The distal colon functions mainly as a storage colon. Its primary role is to store the formed feces before they are eliminated from the body during defecation. Absorption still occurs there, but to a lesser extent than in the proximal colon.
What are the specific mechanisms of absorption in the large intestine? Are they just the same as in the small intestine?
They are similar in some fundamental ways, but there's a key difference in the structure of the epithelium that makes the large intestine exceptionally efficient at conserving sodium against a gradient.
Okay, what's similar first?
Like the small intestine, the mucosa of the large intestine has a high capacity for active absorption of sodium ions, driven by those Na plus K plus ATPase pumps on the basolateral membranes. And just as in the small intestine, this active sodium transport creates an electrical potential gradient across the mucosa, lumen negative relative to blood, that helps drive the passive absorption of chloride ions along with the sodium.
So active sodium transport is still the main engine. You mentioned a key difference in the epithelial cells themselves.
Yes, this is crucial for the large intestine's portion. The tight junctions, the seals between adjacent epithelial cells lining the large intestine, are much tighter and less permeable than the tight junctions found in the small intestine.
Tighter junctions in the large intestine. What does that achieve?
In the small intestine, those relatively leaky, tight junctions allow some back diffusion of ions and water from the pericellular space back into the lumen, especially when the luminal concentration gets low. In the large intestine, these much tighter junctions largely prevent that back leakage.
What's the consequence of preventing back leak?
It allows the large intestine's epithelial cells to absorb sodium ions far more completely, pumping sodium out against a much steeper concentration gradient than the small intestine can manage. It enables the colon to pull sodium levels in the lumen down to very low values, maximizing conservation.
That structural difference in junction tightness is fundamental then to the large intestine's specialized role as the final high-efficiency sodium and water conservator for the body.
It truly enables that last-ditch effort to reclaim almost every last possible bit of sodium and the water that follows it.
And does aldosterone play a role here too? You mentioned its effect earlier.
Yes, aldosterone's effect is particularly strong and physiologically important in the large intestine and the distal tubules of the kidney. Aldosterone significantly enhances the sodium transport capability of the large intestinal epithelium. This is absolutely vital for minimizing sodium and water loss in the feces, especially when the body is facing dehydration or salt depletion.
So aldosterone tells the colon, save all the salt and water you possibly can.
That's essentially the message, yes. Its function in the colon perfectly mirrors its key role in the kidney tubules for maintaining whole body fluid and electrolyte homeostasis.
What about that bicarbonate tech-chloride exchange mechanism? Does that happen in the large intestine too, like in the ileum?
Yes, it does. The mucosa of the large intestine actively secretes bicarbonate ions into the lumen while simultaneously absorbing an equal number of chloride ions via that same anion exchange transport protein we discussed for the ileum.
And the purpose again is to neutralize bacterial acids.
Exactly. This secretion of alkaline bicarbonate helps to neutralize the potentially damaging acidic waste products like acetic acid, butyric acid, generated by the extensive bacterial fermentation of undigested carbohydrates that occurs in the colon.
And water absorption here just follows the absorption of sodium and chloride driven by osmosis.
Precisely. The active absorption of sodium and chloride ions creates the necessary osmotic gradient across the large intestinal mucosa, which then drives the passive absorption of water from the lumen into the blood.
So the large intestine really acts as a vital water and electrolyte recycling plant, ensuring minimal loss. Is there a limit to how much fluid it can handle? What's its maximum absorptive capacity?
Yes, there is a limit. The large intestine can absorb a maximum of about 5 to 8 liters of fluid and electrolytes per day. This is normally far more than the 1.5 liters it typically receives from the small intestine.
But if the inflow is excessive,
If the total amount of fluid entering the large intestine, either due to excessive delivery from the small intestine, like in some malabsorption syndromes, or due to excessive secretion within the large intestine itself, like in infectious diarrhea, exceeds this maximum absorptive capacity of 5-8 lesti, then the excess fluid simply cannot be absorbed. It gets expelled from the body as diarrhea.
which brings us back again to those severe secretory diarrheas like cholera.
Precisely. Toxins from bacteria like Vibrio cholerae or certain pathogenic strains of E. coli can induce the crypts in the terminal ileum and large intestine to secrete massive volumes of fluid, potentially 10 liters or even more per day. This secretory volume completely overwhelms the absorptive capacity of the large intestine, leading to that profuse, life-threatening diarrhea, dehydration, and electrolyte loss.
Beyond its critical role in absorption, the large intestine is famous, or perhaps infamous, for its incredibly dense population of bacteria.
Yes, it's home to trillions upon trillions of bacteria, collectively known as the gut microbiota, particularly concentrated in the absorbing colon. They live in a complex, mostly symbiotic relationship with us, their host.
Do these bacteria contribute significantly to our digestion?
For humans, their direct contribution to digesting our food is relatively minor compared to our own enzymes. They can ferment small amounts of undigestible carbohydrates, like cellulose and resistant starch, that reach the colon. This fermentation produces short-chain fatty acids like butyrate, acetate, propionate, which the colon cells can absorb and use for energy, providing a few extra calories.
So a little bit of energy recovery from fiber.
A small amount, yes. But it's relatively insignificant for overall human nutrition compared to what happens in herbivores like cows or rabbits, where bacterial fermentation is a primary mode of digestion.
But these gut bacteria do synthesize some important compounds for us, don't they? Things we can't make ourselves.
Critically important ones, yes. The bacteria residing in the colon synthesize several essential vitamins, including vitamin K, vitamin B12, thiamine, vitamin B1, and riboflavin, vitamin B2.
Vitamin K from bacteria is particularly important, isn't it?
Yes, the source specifically highlights vitamin K. It's essential for the synthesis of several blood coagulation factors in the liver. The amount of vitamin K produced by our gut bacteria can be quite significant, and importantly, the amount of vitamin K consumed in the average daily diet is often insufficient on its own to maintain adequate blood clotting function.
So our gut bacteria provide a vital backup or maybe even primary supply of vitamin K for us.
Exactly. It's a remarkable example of symbiosis. They get a place to live and food scraps, and we get essential vitamins like K, which helps prevent us from bleeding excessively.
That's a really crucial contribution from our internal microbial residents helping our blood clot properly.
It truly highlights the complex and often beneficial relationship we have with our gut microbiome. Of course, these bacteria also produce various gases as byproducts of their fermentation metabolism,
Which contributes
to intestinal gas, or flatus. Common gases produced include carbon dioxide, CO2, hydrogen gas, H2, and methane, CH4. The amounts and types vary depending on diet and the specific bacterial populations present.
Finally, after all this absorption and bacterial action in the large intestine, what makes up the end product of this entire digestive journey, the feces?
Under normal circumstances, feces consist of about 3/4, 75% water, and 1/4, 25% solid matter.
Okay, mostly water still. And what's in that solid quarter? Is it just undigested food?
Not entirely. It's actually a complex mixture. A surprisingly large fraction, about 30% of the solid matter, consists of dead bacteria shed from the massive colonic population.
Dead bacteria make up almost a third of the solid waste.
Yes. Another 10% to 20% of the solid matter is fat, both unabsorbed dietary fat and fat from bacteria slough cells. About 10% to 20% is inorganic matter, like calcium phosphate. There's only about 2% to 3% undigested protein. And a significant portion, around 30%, is composed of undigested roughage from food, like cellulose fiber, plus dried constituents of the digestive juices, like bile pigments and epithelial cells, that have been sloughed off the intestinal lining during its constant renewal.
It's quite a mix. And what gives feces their characteristic color and odor?
The typical brown color comes primarily from derivatives of bilirubin, which is a breakdown product of hemoglobin from old red blood cells and is excreted in bile. Bacteria in the gut modify bilirubin into compounds called stercobilin and urobilin, which are brown.
Okay, bile pigments determine the color and the odor.
The odor comes from various volatile chemical products generated by bacterial action on the remaining undigested material, particularly proteins and amino acids. The specific compounds responsible for the odor vary depending on the individual's gut flora and diet, but common odoriferous substances mentioned include indole, skitol, mercaptans, sulfur compounds, and hydrogen sulfide, H2S, rotten egg gas.
So even the final waste product provides clues about the complex series of digestive processes, the massive contribution of our internal bacterial residents, and the efficiency of the absorption that happened upstream.
It really ties the whole journey together.
Wow. Okay, we have truly taken a deep dive today into the incredible, often invisible, yet utterly essential machinery of your digestive system. It's quite a journey.
It really is.
We started right at the beginning with the fundamental chemical process of hydrolysis, using water to break bonds, catalyzed by a whole host of specific enzymes tailored for each type of macronutrient.
From salivary amylase and pepsin giving things a head start, to those incredibly powerful pancreatic enzymes doing the main bulk of the work in the small intestine.
And then finally, those brush border enzymes, and even enzymes inside the absorptive cells, completing the job right at the site of uptake, breaking everything down into the simplest absorbable units.
And we saw how the small intestine is anatomically designed with those folds, villi, and microvilli to maximize the absorptive surface area to that astonishing tennis court size.
Absolutely. And how it employs such clever and specialized transport mechanisms for getting different nutrients across the gut lining. That sodium co-transport system is clearly a workhorse.
Definitely, powering the uptake of most sugars and amino acids by leveraging the sodium gradient set up by the Na plus K plus pump.
Contrasting with facilitated diffusion for fructose, and then that completely unique micelle system for ferrying lipid breakdown products to the membrane.
Followed by the reassembly of fats inside the cell, and their packaging into chylomicrons for transport via the lymphatic system, bypassing the liver initially. It's fascinatingly different.
And we didn't forget the crucial work of the large intestine in that final reclamation of remaining water and vital electrolytes, aided by those extra tight junctions and the hormonal influence of aldosterone.
While also benefiting from the metabolic contributions of our bacterial residents,
Yeah.
especially their help in providing essential vitamins like vitamin K.
Yeah. Understanding all these intricate steps, from the molecular breakdown by specific enzymes to the function of individual transport proteins, the role of bile and micelles, and the specialized anatomical structures involved. It really does highlight the remarkable efficiency and sheer elegance of this fundamental biological process.
Hopefully, going into this level of detail gives everyone listening a much deeper appreciation for exactly how your body extracts nourishment from every single meal you eat. It's not just mushing food up, it's incredibly sophisticated biochemistry and physiology.
Absolutely. And, considering that point about the vital vitamin K produced by bacteria in your colon, and the source noting that our dietary intake isn't always enough on its own, it really makes you think, doesn't it?
What about factors that might disrupt that crucial gut bacterial balance? Things like antibiotics or major changes in diet or maybe even stress. What could the broader, perhaps unseen implications be for your overall health and your actual nutrient status, beyond just digestion itself? It's definitely something worth pondering long after this deep dive ends.